First seminar is with Jessica Bell, if you want to come down. So she has been looking at the influence of biostimulants on plant growth and microbial associations of sunflower and strawberry.
Hello. Thanks, Beth. So, what are biostimulants? There are many definitions of biostimulants. The European Biostimulant Industry Council has defined them as diverse compounds, substances, and microorganisms that are applied to plants or soils to improve crop vigour, yields, quality, and tolerance to abiotic stress. The three main categories of biostimulants are seaweed extracts, microbial inoculants, and humic substances. Biostimulants can act via a number of mechanisms, including phytostimulation, where a biostimulant contains or synthesizes phytohormones that influence plant metabolism. Biofertilization, where the availability or supply of nutrients is enhanced. Biocontrol, which involves the increased resilience to pest and disease. And the increased tolerance to abiotic stress. Given that biostimulants have the capacity to improve plant performance, it's not surprising that there's been significant growth in the industry, which is now predicted to reach over $5.5 billion by 2026. That's the global industry, not the Australian industry. On the right, we can see that the majority of biostimulants are applied as foliar sprays, followed by seed and soil treatments. So given the significant growth in the industry, understanding the level of regulation is important. In 2019, the European Union introduced a new suite of regulations requiring a baseline of scientific evidence for all product registration and labelling. Unfortunately, in Australia, we don't have any cohesive regulatory framework. This means that while biostimulants have the potential to add value to production systems, a lack of strong regulation in Australia means that there's continuing variability in product quality and inconsistent results in the field. Which brings me to my project. So the core aims of my project were to test the utility of a mixed microbial inoculant, as the majority of research to date has focused on applying single or limited combinations of microbia, despite an increasing number of multi-species products appearing on the market. Second, as the next generation of biostimulant products look to combine different categories, often under the assumption of the more the better, I thought it was important to look at how biostimulants actually interact when they're combined and identify any potential synergistic or antagonistic effects. And third, there's been limited research to date looking at the potential for biostimulants in soilless production systems, and in Australia, an increasing number of growers are producing important crops like tomato and strawberry hydroponically, so I thought looking at biostimulants for their systems might be of value. To address these aims, I applied a range of biostimulants both as foliar sprays and as a media treatment. The products I applied as a foliar spray were a mixed microbial inoculant containing a whole range of beneficial bacteria and yeasts, a seaweed extract derived from the common brown alga Ascophyllum nodosum, and a fulvic acid solution extracted from leonardite shale. As a media treatment, I applied a soil program that consisted of the same mixed microbial inoculant Microlife, combined with a soil conditioner that contained seaweed extract, humic acid and molasses. Altogether, that gave me an experimental design that looked like this. 8 foliar treatments, so each product alone and in combination, with 2 media treatments, control and the soil program, which was 16 treatment combinations altogether. This design was repeated across 2 pot trials in the greenhouse, the first using dwarf sunflowers in nursery grade potting mix, and the second using the strawberry cultivar Toscana F1 Rose. Strawberries were grown hydroponically in coconut coir and fertigated with Hoagland solution. My approach to the project was twofold. The first component was to understand how applying biostimulants influences plant microbial associations. For a microbial inoculant to act effectively, it must be able to colonise and persist on the leaf surface and in the growing media. One technique for understanding whether colonisation has occurred is to test for biofilm formation. So I wanted to establish whether Microlife, applied as a foliar spray, was able to initiate biofilm formation on the leaf surface. I also wanted to understand whether the addition of other biostimulants would assist or inhibit biofilm formation. To answer these questions, I performed a series of crystal violet assays at 2, 6, 10 and 14 days after treatment. The results from both trials demonstrated similar trends. The control, which was sprayed with tap water only, consistently had the least biofilm formation. Plants treated with Microlife and Microlife combined with fulvic acid had more biofilm formation, although not significantly at every time period. But interestingly, it was plants treated with Microlife combined with products that contained seaweed extract that had the consistently highest biofilm formation at all time periods in both trials. So what are the takeaways? Yes, Microlife was able to form a biofilm, but this was significantly enhanced by the addition of seaweed extract. While not directly addressed in this trial, potentially the seaweed was acting as an adhesive, sticking microbes to the leaf surface, a humectant, preventing desiccation, or a stabiliser, providing nutrients while the microbes established. What we can't tell from this data is which microbes from the consortia actually formed the biofilms, and does this translate to enhanced protection via biocontrol or plant performance? I also wanted to investigate whether applications of the biostimulant soil program would influence the microbial population density in the substrate. I assessed this via a series of most probable number assays. In the sunflower trial, the results suggested that yes, the treatment with the soil program increased the microbial population density in the potting media, compared to both the before sample and the control. In the strawberry trial, I asked the same question again, as well as one additional question that emerged from a hypothesis that high soluble nutrients in the fertigation solution would inhibit microbial establishment in the coir substrate. To test this hypothesis, the trial was divided into two phases. The first phase saw plants receive 100% concentration of Hoagland solution via fertigation, and the second reduced the fertigation concentration to 30%. And here are the results. We can see that at the end of phase 1, when plants had been fertigated with 100% concentration of Hoaglands, the soil program was not able to significantly increase the microbial population density in the substrate. However, at the end of phase 2, after the concentration was reduced, the soil program was able to increase the microbial population density. So the takeaways from this section of the project were that the soil program was able to increase microbial population density, but only under low nutrient conditions. High soluble nutrients in fertigation solution did seem to inhibit microbial proliferation in a hydroponic system. What we can't tell is whether the microbial inoculant or the other components of the biostimulant program, so the seaweed, humic acid, molasses, drove increases in the microbial population density. Does this actually translate to enhanced plant protection by biocontrol and plant performance? And most importantly, could the benefits of microbial inoculation actually justify reducing the concentration of the fertigation solution in a commercial context? So, to the second portion of the trial. First, I looked to examine whether biostimulants influence plant growth and whether there are any synergistic or antagonistic effects when applying biostimulants in combination. To answer these questions, I collected data in both trials looking at a range of plant growth, health and yield parameters. Of all of the parameters, only the following returned significant results. And further analysis revealed that for those characteristics that were influenced, it was only foliar treatment with biostimulants that produced significant effects. Unfortunately, I don't have time to go through all of the results from my project, but here are some of the key findings. So in the sunflower trial, plants treated with seaweed extract combined with fulvic acid had the greatest number of leaves on average, significantly more than plants treated with all other treatments except for seaweed alone and the control. Interestingly, plants treated with Microlife, so the microbial inoculant, produced the fewest leaves on average. In the strawberry trial, again, seaweed and seaweed combined with fulvic acid were the top performing treatments, producing significantly more berries than plants treated with Microlife and the control. It's also interesting to note that on average, plants treated with the combination of all three biostimulants, microlife, fulvic acid and seaweed extract combined, produced significantly fewer berries than plants treated with seaweed alone, highlighting a potential antagonistic effect. A similar pattern was observed for the total yield per plant. Seaweed was the top performer, followed by seaweed combined with fulvic acid. Again, the control had the lowest average, followed by microlife alone and the combination of all three biostimulants. So what are the takeaways for plant growth? In both trials, the majority of plant growth characteristics were unaffected by treatment with biostimulants. The multicraft soil program had no significant effect on plant growth in either trial. However, treatment with seaweed extract and seaweed extract combined with fulvic acid were the top performers in both trials. Again, this wasn't directly assessed in this project, but the potential mechanisms may have been phytostimulation or biofertilisation, or some combination of the two. So, in conclusion, the results from my project indicate that seaweed extract is a reliable and effective biostimulant, and it is able to improve plant growth, yield, and potentially enhance biofilm formation when combined with a microbial inoculant. Second, as mixed microbial inoculants are inherently complex, with interspecies dynamics within the inoculum, crop compatibility, existing site conditions, mode of application, product stability, a whole range of factors influencing their effectiveness. More work needs to be done to work out when they're actually suited to application in the commercial context and when they might offer the most benefit. In terms of applying biostimulants in combination, there's definitely the potential for additive benefit. However, it's not always necessarily the more the better. And finally, stronger regulation in Australia would be a positive thing to improve reliability and improve reliability and return on investment for growers. Thank you.
I want to say, so we definitely can have questions in the room. Whether we can have questions on the online seems a little bit vague. So I'm sorry if you're online and you want to ask a question, you can send them to me by email and I will ask them another time. But have you got any questions in the room for Jess? So Jeff is asking whether you see any differences between sunflowers and why.
I suppose I didn't directly select those two as a comparison. I selected sunflowers because, especially the dwarf sunflowers, because they were fast growing, easy to grow in pots, so it was more a practical choice. And then strawberries were chosen because I hoped that the results might be of more relevance to industry. So sunflowers was, more of a practical choice for an honours project, and strawberries for industry relevance and personal interest. Yep. Oops.
Googled? Googled, yep. So what Dougal's asking, for those who are watching online, they can't hear the people in the audience, Dougal's asking what the mechanism might be of seaweed extract in terms of fruit number and leaf number, is that? Yeah.
So none of the data that I collected gave me an insight into the mechanisms, but from reading, from what I understand, seaweed extracts contain a whole range of phytohormones, so auxins, cytokinins, as well as micronutrients and alginates. And those different components can influence plant growth in different ways, which other studies have looked at those mechanisms. So in this study, especially when applying seaweed extract as a foliar treatment, there's the potential that the phytohormones are influencing plant metabolism, but also that the micronutrients are being absorbed through the leaf surface, and that might be stimulating plant growth. That was something I sort of thought we might find, that combining fulvic acid with seaweed extract would have a beneficial influence on plant growth, because fulvic acid has a very small molecular weight and size, and other papers have said that it's able to be absorbed through the leaf tissue and can actually chelate micronutrients and act as a sort of carrier, carrying them into the plant system. But yes, I didn't directly look at that.
And Peter?
Yes. Optical density. So a crystal violet assay is basically a stain that binds to the extracellular polysaccharides created by living biofilms. So you stain it with crystal violet and then solubilize that with ethanol, put it in a 96-well microtiter plate and into the spectrometer. And so then that gives you the optical density, gives you sort of a comparative measure of the amount of biofilm in the sample.
And I think we'll have one last one, maybe from Kathy. So what Kathy's asking is whether biostimulants might be more useful in modified environments than in the field.
Different.
Is there a difference between how they interact?
So that I don't prattle on for a long time, I'll focus on one, so microbial inoculants. In the field, applying a microbial inoculant, say, to the soil, obviously the soil biology in a field soil has an incredible array of microbial diversity already. And so there's big question marks as to how much, how frequent, what mixture of microbes could you apply to a field soil that would actually make a difference, and could they compete with the native biology in the soil? Whereas in a soilless production system, in, say, coconut coir, it's not sterile, but it also doesn't have a huge diversity of microbia in that substrate. So one of the starting questions in my mind was, would there be a benefit to inoculating that substrate and making it more like a living soil? Could you make it more like a living soil, and would that benefit the plants growing in the substrate? But yeah, the results of the project, there's more work needs to be done on that.
Thank you very much. I'd like to thank Jess again.